Picture this. A happy toddler suddenly spikes a fever. Tiny blisters pop up on their hands and feet. Then, almost overnight, they can't stand. A harmless-looking gut virus has slipped into the brain. Here's the head scratcher. That very same virus can hang out in the child's intestine for weeks without making a peep. How does it pull off that vanishing act? Today, we're diving into the brand new mini gut on a chip that's finally giving us answers. First, why did scientists even need a new model? Let's zoom out for a second. Virologists have been juggling two lousy choices. Choice 1. Infect animals. You get the whole body, but mice aren't tiny humans. Choice 2. Grow a flat sheet of human cells in a dish. Human, yes, but about as simple as a paper napkin. It's like trying to learn orbital mechanics by watching a playground swing. You see motion, sure, but you miss the cosmic rules that matter. Enter enteroviruses, especially EVA71. These bugs exploit everything those older models ignore. The constant birth and death of gut cells, rivers of mucus, the squeeze of peristalsis. To watch that ballet in detail, researchers in Tokyo built an intestine-on-a-chip. Think of it as a living slice of human gut under glass, with knobs and dials for flow, nutrients, and even mechanical stretch. In other words, a backstage pass to the virus's playbook. Signpost: how does this chip actually work? Scientists seeded one side of the microfluidic chip. Picture two parallel channels separated by a porous membrane, with primary human intestinal epithelial cells. They let those cells grow until tiny finger-like villi and a cozy mucus blanket appeared. Only then did they introduce EVA71 into the lumen side, the side that would normally face whatever you just ate for breakfast. The opposite channel stayed virus-free but fully perfused, so researchers could keep the tissue healthy while filming the infection in real time. Now, here's where things get fun. Over several days, they spotted clusters of infected cells sitting right next to brand new, squeaky clean cells that had just crawled up from the stem cell layer. As the older, infected cells naturally peeled off into the gut lumen, part of normal turnover, the virus simply hopped on board the younger neighbors. Picture a relay race. One runner hands off the baton just as they step off the track. The infection never has to sprint, it just keeps passing the baton. Signpost: what about the immune response? When the team ran transcriptomic profiling, fancy word for reading which genes are turned on, they found something odd. The chip dialed down type 3 interferon, the gut's local alarm system, way more than traditional 3D organoids did, even though viral RNA levels looked the same. Two possibilities popped up. 1. The tissue's complex architecture physically mutes the alarm. 2. The virus is better at cutting the wires when the environment feels more like home. To test that, the researchers sent tiny pulses of interferon through the basal channel and watched how far the danger message spread. Because the infection stayed stable for weeks, the chip turned into a long-running observatory. Chronic-like states set in, then add experimental drugs, or use CRISPR to dial the viral receptor SCARB2 up or down. It's less like a lab bench and more like an ecosystem terrarium. You tweak one variable and watch everything shift in slow motion. Quick example, swap EVA71 for a cousin that also uses SCARB2 and see if it plays by the same rules. Or seed chips from different donors and look for genetic quirks that tip the balance between silent carriage and severe disease. Layer in friendly gut bacteria and ask whether certain microbes crank the viral volume up or down. Little by little, you start mapping selective pressures that breed either stealthy, immune-evasive mutants or fast-spreading hotheads. So why should you care? 3 big reasons. 1. Kids first. Silent, long-term gut infections often precede the sudden, scary neurologic cases we see in clinics. Catching that slow burn could save lives. 2. A reusable test bench. Because the same chip can be infected over and over, it's a low-risk, high-control way to screen antivirals or tweak vaccine candidates before moving into costly human trials. 3. A bigger trend. Organ-on-chip models are popping up everywhere. Lungs, brains, even placentas. They're cutting down on animal studies and opening windows into human biology we simply couldn't crack before. Let that sink in for a moment. Now, Before we wrap, let's bring it home with a quick thought exercise. Imagine you had one afternoon in this Tokyo lab. What's the first thing you'd test? Viral entry tricks, immune signaling, or maybe drug timing? And why would that matter for real patients you know? Jot it down. That single question could shape your next grant, your next collaboration, maybe even your next breakthrough. So, here's what we covered today. We opened with a simple riddle. How can the same enterovirus be harmless in the gut, yet devastating in the brain? We saw how the intestine-on-a-chip bridges the gap between clunky animal models and oversimplified cell dishes. We learned that EVA71 uses the gut's natural cell turnover like a relay race, quietly staying one step ahead of immune alarms. We discovered that the chip's controllable environment lets scientists tweak flow, nutrients, and immune cues to spot viral weak points. And finally, we zoomed out to the bigger story. Organ-on-chip technology isn't just a gadget, it's a new lens on human disease. Here's your takeaway. The next time you hear "on a chip" in the news, don't tune out. Ask yourself, what hidden corner of biology is that chip about to light up? And more importantly, how could that new window change the way you care for patients, design experiments, or simply understand your own body? That curiosity is your superpower. Use it. Until next week, stay curious and keep asking the questions that push science, and all of us, forward.